So now we're going to talk a little bit about meiosis. And in order to do that, I just want to discuss a little bit of detail about chromosomes and what their structure is. Okay, so chromosomes in a nucleus usually exist in pairs called homologous pairs. And basically what it is it's kind of like having the same sort of agenda on each one. Each one's coding for the same information, but because one of them comes from the mother and one of them comes from the father, the information might be the same. So for example, they could both be indicating eye color, but one of them will code for brown eye color and one will code for blue eye color. And these genes that are sort of complementary but different are called alleles. And that's how homologous chromosomes pair up. So the chromosomes that are similar in shape and have similar information coded on them will basically be paired as homologous chromosomes. And that's how chromosomes exist in a normal cell. After DNA has undergone replication, what you're going to get is you're going to get an exact copy of each of the homologous chromosomes within the pair. So even though it looks like this, ultimately that's still just one pair of homologous chromosomes, one from the mother and one from the father, and they each consist of two identical strands of the same thing, which are called sister chromatids. So each of those thick strands there is called a chromatid, and they're connected at the middle, over here, by a centromere. Okay, so that's what happens after replication. So it can be a little confusing because sometimes this is represented as a homologous thing, but essentially what you have to remember is that homologous chromosomes look like this, and then when they replicate, that's when they look like this. And the reason why we usually see pictures of chromosomes looking like this, as opposed to just kind of squiggly and around, is because this is the only time they're visible on the microscope. Once they've undergone replication and they're getting close to dividing, they condense very closely together to form very thick strands, just to make separating and everything easier. So that's how they appear on the microscope, and therefore that's how they're usually drawn. Okay. Oh, and then this sort of cluster where you've got a homologous pair, made of two sister chromatids. This is called a tetrad, because if you imagine that you've got information for eyes, it's going to be that information on 4 times. So tetra means 4. Okay. So in terms of identifying cells, you just have to know the difference between diploid and haploid. So diploid is any cell that actually contains a homologous pair of chromosomes, i.e. one from each parent, as we've discussed. And that's basically all the cells that are functional in the human body to keep you going. The only cells that are not diploid are the reproductive cells, the gametes that are in your eggs and in male sperm. The reason so haploid means they do not have a homologous pair, so they'll only have one of each of these. There'll be one strand coding from the mother, one strand coding from the father. Okay, and the reason why that's the case is because when fertilization happens, in order to create a baby, what you're doing is you're combining the genetic material from the mother and the father. So you want to be able to provide that, but you don't want to double the genetic material in total. So if there were homologous pairs, you would be giving 2 from the mom and 2 from the dad, and you would actually wind up with 4 chromosomes, which isn't really functional. So basically, the reason why haploid cells exist is so that when fertilization happens, you have a proper set of homologous chromosomes in the child. Okay, another word for reproductive cells, by the way, is gametes. Okay, yeah. So basically, in summary, that's what meiosis does. It'll take one diploid cell, as in one cell that has a homologous pair, usually within a reproductive organ. It undergoes 2 divisions so that ultimately you end up with 4 cells, each with a single chromatid in it. So they are haploid cells, and we'll go through that just now. But basically what this is called when you're moving from a diploid cell to a haploid cell is reduction division, because you're reducing the homologous chromosomes in there, the number of chromosomes ultimately. Okay. Right. Okay, so this is the overall steps of what happens in meiosis. I'm going to talk through this in a second, but what I want to focus mostly on is what happens to the chromosomes. Essentially, the steps that happen are very similar to what happens in mitosis, and there are only specific differences. Okay, so I am going to, here are the notes. I'll talk about that in the next video, specifically about the chromosomes, but here's just the overview of what happens during a meiotic division. Okay, so meiosis also starts with an interphase area where it's got a cell that is a diploid cell. In this, I'm following what happens with two sets of chromosomes as opposed to just one, just so that you can also see the pattern of what's happening and see how similar it is in terms of the mitosis phases. Okay. So first the interphase happens, it's much the same, it grows and DNA replicates. And then what you've got is you've got two sets. You've got a first meiotic division and a second meiotic division, okay, or meiosis 1 and meiosis 2. So in meiosis 1, we've got all the same steps that we have in mitosis. So we've got prophase 1, where the copied DNA sort of gathers together into its homologous pairs. You can see the same stuff is happening. The spindle is forming, the nucleus is disintegrating and everything. I'm not going to talk through all of that throughout this, though. It's the same stuff happens in each phase as it does in mitosis in terms of the mechanics of the division. Okay, but what's really important is that in prophase 1, your homologous pairs actually cross over, like the legs and stuff cross over and twist together, and the point where it does that is called a chiasmata. Okay, and then what it does is it actually swaps genetic information, so that some of the information that's on the blue chromosome is now on the pink chromosome. I'll talk a bit more about that just now in detail. The point is it's swapping genetic information. So that's the important thing that happens in prophase 1 of meiosis. Okay, in metaphase 1, there's something called independent assortment. Basically, that means that these chromosomes are separated. Your homologous chromosomes are separated. In mitosis, they are not separated. It's the chromatids, the exact copies, that are separated. But in this case, we're actually only separating the homologous chromosomes, and those separations happen at random. So I actually shouldn't have drawn it like this. What would have been a better indication would have been if on this side one of the mostly blue ones came and on this side one of the mostly pink ones came, because you've got 23 pairs and there's no like the father ones come to the left, to the right, and the mother ones go to the left. It's just random which ones go where. Okay, so that's the important thing that happens in metaphase 1. And in anaphase 1, these again, homologous chromosomes are pulled apart, not the copies, not the chromatids. And then you've basically got the telophase and cytokinesis. And you can see that already these chromosomes are all looking a little bit different. Okay, so basically what's going to happen is now you're going to have two cells. Okay, these cells are not genetically identical, and they are now haploid cells, because even though the chromosomes still have their exact copies, they're no longer in their homologous pairs. And when I say exact copies, I say they're not exact anymore. Okay, so now we're going to enter into the second part of meiosis, the second meiotic division. And from this part, it's pretty straightforward. It just kind of follows the steps of mitosis. The chromatids all line up, they get separated, and pulled to different cells. And then ultimately what's going to happen is that each cell is going to be a little bit different. They're going to all of the genetic material is going to be slightly different in each cell. And then you're going to wind up, again, just through the same steps, to 4 haploid cells, each with completely different genetic material.